you. Can everyone hear me okay, even in the back? Okay. I have a fairly loud voice, and if I'm too close to the mic, it gets muffled, so I don't want to make it hard to hear me. Well, as Kathy said, my name is Amy Pollpeter. I work for the Iowa Division of Criminal Investigation. Is there anyone else in the room who's not a forensic nurse? All right, or forensic medical practitioner. I just kind of wanted to know what I was speaking to, if there was anyone besides nurses that were in the room. I'm going to talk a little bit today about some of the new emerging technologies in DNA, as well as some other just DNA issues that have come up as I've been working with our SARTs in Iowa. That's our disclaimer slide. So after I talk about those new emerging technologies, I also want to talk about the dreaded C word, contamination, and that it's not always as bad as we may think it is when you hear that word. And also the crime lab SART relationship and how important that relationship can be. So I'm going to start off with some new and what's really not so new emerging technologies in DNA that affect sexual assault and forensic nursing in general. But first I want to start with a little bit of a review on DNA testing, because I don't know what all of you guys know about DNA and DNA testing and how it's based. The nurses in Iowa have to listen to me rattle on for about an hour about the science behind DNA testing, as well as what we do at the crime lab. I promise this won't be that long. It's one slide, so it's fairly painless. Polymerase chain reaction, or PCR. How many people in the room have heard of this term? Yay! That's good. Is there anyone who would be willing to volunteer what they think it means, or an explanation of it? Anyone? No one? Everyone's shaking their heads no? Well, at least you've heard of it. That's great. Polymerase chain reaction is what the main biology of DNA testing is based on. It's a technique that's not used just for forensics. It's used for medical facilities. It's used in plant breeding programs, zoo breeding programs. It's used all throughout molecular biology. But basically, it's a Xerox machine. It's a chemical Xerox machine. It makes lots and lots of copies of a small amount of DNA. This is what enables us to take a DNA stain that's very, very small, maybe 1 millimeter or 2 millimeters in diameter, and get a DNA profile that's strong enough that we can actually detect it and interpret it. STRs, or short tandem repeats. How many people have heard of this? Okay, still quite a few people. Anybody want to try to explain what they are? No. There are forensic scientists who don't want to explain what STRs are, to be honest. But a short, basic definition is there are areas of our DNA that's what sometimes is referred to as junk DNA, but really it's non-coding DNA. It's DNA that's in an area that we no longer use to determine our physical features or development or illnesses or anything like that. So it has no real function anymore. I think it's very analogous to a blueprint. Our DNA is kind of like a set of blueprints for a contractor. When they're building a house, the blueprint tells them where to put rooms, how big the rooms are, all of those types of information. Our DNA does the same thing for our bodies. The short tandem repeats that we use for testing are in an area of the DNA that is very analogous to the white paper that a blueprint would be printed on. It doesn't really provide any information by itself, but it's part of the structure of that blueprint. And these short tandem repeats don't provide us any information. The only information we get from DNA testing on nuclear DNA testing is whether or not someone is male or female. That's the only physical information we get. Everything else is just a random number, essentially, for what their DNA profile is. How many people know that your DNA is a mixture of mom and dad? That's why you are kind of a conglomerate of traits from both of them. I know that both of my two sons have some traits like me and some traits like their father, my husband. They probably have some traits like my dad too, because I got them from him. There's 13 CODIS core locations. Most of the DNA kits that we test at right now, I think that's actually my next bullet. Oh, it's not, okay. Most of the DNA test kits right now being used test at 15 locations. And in molecular biology, just kind of like nursing, we have our own lingo, we have our own vocabulary. For some reason, they can't use the term locations. They use loci in genetics. It means location in the genome. We test at 13 of them right now. Most kits actually test for 15, 13 are required to enter into the national DNA database. How many people have heard of CODIS? Yay, that's on the news all the time, so everyone knows about CODIS. CODIS is the national DNA database. I'm going to come back to the statistics in a minute. And basically, CODIS is actually a conglomerate of databases. It's databases for forensic samples, which means this is samples from our evidence. So male DNA profile developed from a vaginal swab in a sexual assault kit, a blood stain at a crime scene. Those are the forensic samples. They have offender samples. These are people who are required by law to submit a sample to the DNA database because of being convicted of a specific crime. What crimes they have to submit for is dependent on each state. It changes by state. Some states do. Almost all states do all felonies now. Some do some misdemeanors. It just depends on what their state legislation is. And it now also contains arrestee samples. Arrestee samples I'm going to talk about a little bit more later on in the presentation, so I'm not going to get into it too much right now. Now I also have a bullet up here about statistics. If you've ever listened to a DNA analyst testify in court, first of all, I'm sorry, but because it can be rather technical at times depending on what the lawyers decide to ask. But if you've ever listened to them testify, you're going to hear anytime they say that a DNA profile developed from the evidence matches a specific person, they're going to give a statistic that follows that. And that's required by the Federal Bureau of Investigations. It's basically a way for the trier of fact, either the judge or the jury, to determine the weight of that DNA profile. How likely is it that someone else in the random population could have that DNA profile by chance instead of whoever we said it matched, generally the suspect? In the news, you may have heard lately about a problem with the statistics that DNA analysts have used. About in 1999 and 2000, the Federal Bureau of Investigation, and I'm not picking on them, they have readily admitted that they did this, they published data that the entire country based their statistical programs off of. There's several professional computer programs that are based off it, as well as in-home ones, Excel spreadsheets, whatever the laboratory used to determine their statistics, they based it off this data published by the FBI. In an audit earlier this year, the FBI discovered that that data was hand-entered and there was errors in it. That's never a good thing. So all of what we were basing our statistics on had some flawed errors. Now luckily, it didn't change the statistic that was reported by a lot, but it could change it by a small amount, and this affects every case worked in the last 15 years. So it affects a lot of cases for every crime lab in the United States. Most of the crime labs had to immediately either deal with this by deciding how they were going to address DNA reports. The state of Iowa, we had an audit coming up, and our audits are very rigorous, and so in order to not get dinged in that audit, we immediately suspended reporting all DNA samples until we could fix our statistical program with the amended data that FBI sent out. We hope that that data is correct. I was just in court on Monday, and of course the defense attorney is like, well, they messed up before. Why do you trust them now? Well, because I have to. I mean, I can't go to the FBI and check their work for them, so I'm assuming that their work has been double or triple and quadruple checked. But so you may hear about that in your states, in the news, in court cases, things like that. For the most part, it's not going to affect the results. If you got a full DNA profile of a suspect, or if the DNA analyst obtained a full DNA profile of the suspect, the statistic is going to be so high that it's not going to change. But if they had a very partial profile, which means they only had results at maybe 10 of those 13 locations, or 7 of those 13 locations, it can affect the statistic. And a lot of defense attorneys are now looking at old cases. They're going to be requesting state labs to recalculate the statistics in those cases. In some cases may end up getting appealed or going back to court because of it. So I wanted to bring that up. It's kind of a news item about DNA right now. Which is never good when it's a negative news item. We don't like that, but unfortunately the good stuff that we do usually doesn't get reported. So the first new or emerging technology is expanded STR loci. So I just talked about the fact that in order to put a sample into the national database, we need to have results at 13 locations. Most of our kits test at 15 locations. Now, they're moving to kits that test at 24 locations instead of 15. The reason why they're doing this is because it's more discriminatory. It's basically going to give more power, larger statistics. It's less likely that you're going to have some sort of a match by chance or by coincidence between two people. I don't know that I've ever heard of a match happening between two people, but I've heard of very close profiles, even in unrelated people. It is the same process as our current kits that we use, it's just more loci. As technology has expanded and has grown, they've been able to add in more locations, more genetic markers. This is a good thing, except it's a lot of work for the laboratories to have to reintroduce new technology into their labs. The other thing this does is it allows international searches. With the 13 CODIS core loci that we've been working with, only about 7 of them were used by the European countries. There's hundreds of STR markers that can be used for forensics. And when they were first developed in the infancy of DNA testing, Europe chose a different set of markers than what the United States did. I don't think they were thinking at the time how much this would affect searches later on, but it ends up that we couldn't really compare the DNA profiles that we obtained in the United States with DNA profiles from Europe. And especially in some of the terrorism cases and global crime, this has become much more important lately. It also reduces those CODIS matches or hits by chance. In CODIS, if two profiles hit on each other, it means that they are either exactly the same or very similar. And the more locations you test at, the less likely it's going to be that two people are going to match. Think of it as if you have a set of dice. With your DNA, because you have half of it from your mom and half of it from your dad, if you're using a dice example, it'd be like holding 2 dice. And if you were to throw them one time and get, say, a 1 and a 2, you want to find out how likely it is to get a 1 and a 2 if you throw those dice again. Well, it's 1 out of 6 times 1 out of 6, which is 1 out of 36. However, if you were to say, I want to see what happens if I roll this dice 8 times and write down what I got each time on each dice, and then I want to roll it another 8 times, and how likely is it I'll get the exact same answer on each of those next 8 times? You start multiplying those probabilities together, and it becomes much less likely, or a much lower probability that you're going to get the same answer over and over. So with 15, it was pretty unlikely that we'd get the same DNA profile. Now with 24, it's even less likely. And I wouldn't be surprised if in 10, 15, 20 years, however long it takes technology to catch up, they're going to expand it again. Because at that point, there's going to be even more profiles in the national database. All of the labs are required to use these expanded loci by the beginning of 2017. That's not that far away when you have to do a full validation project in your laboratory and keep up with all your casework. So all of the DNA labs right now are trying frantically to get these new kits validated so that they can use them when the CODIS switches in 2017. And like I said, the CODIS core, the CODIS always seems to pick a slightly lower number of locations than what the kits are testing. So when the kits were testing 15 locations, they required 13. They had 13 that they said were their core loci. Now that the kits are going to be testing 24, they've picked 20 that are the core loci. But these 20 loci or locations are used by both the United States and Europe. So they're going to be the same, and we'll be able to do searches in international countries as well. Why STR testing? This is a technique that's really, it has another name. It's called male-specific DNA testing. Sometimes the media tends to refer to it as that. It's really not new. It's been around for probably 10 to 12 years. I think laboratories started using it about 10 to 12 years ago. It's basically testing genetic locations. Instead of the locations they test at being spread across the entire genome, it's all on the Y chromosome is what these tests test for. Well, what's unique about the Y chromosome? It's only in men. Females do not have a Y chromosome. Females are XX, men are XY, if you go back to genetics classes. And so that's why it's called male-specific. Basically, it ignores female DNA. This can be really useful if you have, say, a victim who has either waited maybe 4 or 5 days before she came to the hospital after being assaulted, or maybe they've showered. They don't have a lot of sperm left in their vaginal cavity, but they have a few. Unfortunately, when you swab that area, you're also going to get a whole lot of the victim's own DNA. And so it can be difficult for the crime lab to determine that male DNA profile because it's mixed in with a whole lot of female DNA. And our testing is really sensitive, and it's gotten more sensitive over the years, but it still has a limitation. It still has a limit where once you get so much female DNA, it just swamps our chemistry kits, and it doesn't work anymore, and we don't get the male DNA profile. So this ignores the female DNA. It allows us to really zero in on that male DNA, which is what we're looking for in a male-on-female sexual assault. One issue with YSTRs is that they are paternally inherited. Because they are on the Y chromosome, they are inherited completely from the father. So any guy is going to have the same Y chromosome DNA as his father did. And brothers will have the same Y chromosome. So it's not as identifying. It's not going to necessarily identify your suspect, but it's going to help you narrow it down. Hopefully, law enforcement techniques and investigative techniques can rule out the possibility of other people in the family being available to commit the assault, because the father and all his male offspring will have the same profile. Now, there can be mutations, but mutations on the Y chromosome aren't super common, so a lot of times you may go several generations without seeing a mutation. So like I said, it's not as discriminating. It's not identifying. The other problem is there is a Y DNA database or a Y profile database, but it's not as large, and because the profiles can be very similar, are exactly the same between males of the same family, and even sometimes males of two different families can still have the same Y chromosome profile. It hasn't expanded to the point or evolved to the point where it's different yet. And it probably won't in any of our lifetimes. So there can be some searching limitations. It becomes really important to have a suspect in the case. A lot of laboratories don't do YSTR testing if there's not a suspect at this time, because they can't really search the DNA profile. So that's one limitation with YSTR testing. Mitochondrial DNA testing. So mitochondria, if you remember back to cell biology, are the little bean-shaped parts of your cell. They're oftentimes called the energy or powerhouse portion of the cell. It has DNA in the mitochondria. The rest of the DNA that we're looking at, that I've been talking about, is in the nucleus of the cell, that brain portion of the cell in the center. But the mitochondria is the only other part of the cell that has its own DNA. This type of testing is also not new. It's been around for probably 10 to 15 years as well. But it's most useful for human remains or mass disasters. Because this type of testing is actually maternally inherited. That means that it comes from all of your mitochondrial DNA comes from your mother. My older brother and I, even though we're different genders and everything, we have the exact same mitochondrial DNA, and it will match my mom. This can be useful in human remains and mass disasters because oftentimes when someone goes missing or when a mass disaster happens, the family members of those victims may not have a DNA profile on hand of their missing loved one, unless they happen to know a forensic scientist. I know all of my family members' DNA profiles, but most random people aren't going to have that. And sometimes it can be difficult to find something to use as a known sample for that missing person, if we find any human remains that we want to compare it back to. With using mitochondrial DNA, all we have to do is find someone who's maternally inherited, a brother, a sister, the mother, someone like that who's alive in the family that we can use as a DNA profile. Like I said, because it's maternally inherited, just similar to YSTRs, it's not as discriminating. It's not going to identify a single person, but it can be used to help either rule people out or narrow down who you're looking at. Very few crime labs perform mitochondrial DNA testing. It requires an entirely different process than the rest of the types of forensic DNA testing. Most of the private labs will perform it, but the state crime labs, they don't have the space or the equipment or the money to perform a mitochondrial DNA testing. It is used occasionally in criminal cases. I've had it used in a couple of my cases where they needed to get DNA from hair. One thing about hair is the only time we can get STR DNA from the hairs is if the hair is forcibly pulled from the body, which means it has to have a root attached to it. If the hair is naturally shed or falls out and then transferred during a crime, we won't be able to get a DNA profile